Yeah, so Cartesian is a pioneer in RNA cell therapy. So we are a clinical stage biotechnology company based out of Gaithersburg in Maryland. We're a fully integrated company in that we do discovery here in-house. We do clinical operations in-house, but we also have our GMP manufacturing facility here and our own quality management systems. So that level of integration has allowed us to matriculate assets from preclinical development to clinical development relatively quickly, so that we currently have 3 assets in clinical development across 3 different disease categories. And then another handful, 3 of preclinical programs that we're working on in our discovery and translational group. Yeah, so RNA cell therapy is a new therapeutic category that is born out of a marriage between the conventional RNA therapeutics and conventional DNA-based adoptive cell therapy. Conventional RNA therapeutics are basically the COVID vaccines, right? So an RNA molecule is engineered into a nanoparticle and administered into the body directly. And conventional DNA-based adoptive cell therapy is your typical conventional CAR-Ts, for example, where you have cells, immune cells, that are engineered with DNA that integrates into the cell's genome. So what we do is take the best from both of these parents into this new disease therapeutic category. So we take the RNA from the conventional RNA therapeutics, and we take the immune cell from the conventional adoptive cell therapy to create this new therapeutic category called RNA cell therapy. And there's some distinct advantages of this approach relative to its parents. And so compared to conventional RNA therapy, RNA cell therapy allows us to be able to use the cell as both a factory for producing as well as a vehicle for delivering a combination of RNA therapeutics right to the site of disease. So we can be much more precise in terms of how we home our RNA therapeutics right to the disease. We also have much less of an issue with immunogenicity. So one of the big issues with RNA therapies and why they're so good as vaccines is because they induce the immune system to activate. But that's a good thing for vaccines, but not a good thing for therapeutics, because then your therapeutic gets cleared up. When you use the cell as a delivery vehicle, immunogenicity is much less of an issue. Compared to conventional adoptive cell therapies, RNA cell therapy is just safer because RNA has a half-life, it has a beginning, a middle, and an end, and you can very specifically control your the life of your therapeutic inside the body, because RNA doesn't get permanently and irreversibly get integrated into the cell genome. And so as a result, we have a lot less toxicity, both short-term as well as long-term, and the risks of transformation, these cells turning into a cancerous cell, are negligible to nil. It's also a lot less expensive to use these cells in these ways. And we also don't have a cargo limit, so we can theoretically engineer many, many different RNA therapeutics within a single cell and use the cell as the delivery vehicle right to the site of disease. So a new therapeutic category, and we're quite excited to be right at the cutting edge of it. Myasthenia gravis is one of our clinical indications we're going after as a representative autoimmune disease. It's really a classic autoimmune disease in that pathogenic autoantibodies attack a particular tissue in the body to cause the disease. And in myasthenia's case, those pathogenic autoantibodies attack the junction between the muscle and the nerve, the neuromuscular junction, and prevent appropriate signaling in the muscle and cause muscle weakness over time. And a lot of patients have the localized form of this, where it just affects, the eyes. It's called ocular myasthenia. And then there's a good fraction of patients that have the systemic form, where they're weak throughout. Before the advent of steroids, a third of patients died as a result of this disease. And even today, the kinds of therapies that are used, these toxic immunosuppressives, are often cause the side effects that are worse than the disease itself. So there's an urgent need to develop new therapies to address this disease. So what we do is use one of our products, Descartes-8, to address this disease. And the way we do it is we engineer these cells, these T cells, with a molecule called the chimeric antigen receptor, a CAR, that binds to a protein that's expressed in the long-lived plasma cells in the body that produce the pathogenic autoantibodies that go and bind the neuromuscular junction. So it stands to reason, if we can kill the long-lived plasma cells that are producing these pathogenic autoantibodies, that we can address this disease right at the source and get rid of the disease right at the source. And that's what these CAR T cells do. The fact that we're engineering them with RNA instead of the conventional DNA way of engineering these CAR T cells makes all the difference, because we can control the life of these cells in the body. They're a lot safer. And as a result, we can go after an indication such as myasthenia, when virtually all CAR T cells out there are relegated to having to target these very end-stage severe patients with advanced cancer. And the reason is because they're often so toxic that it doesn't make sense from a risk-benefit perspective to use these types of conventional adoptive cell therapies to go after anything other than advanced cancer. By making our therapy safe, we're able to go after diseases not only in early stage frontline oncology, but even beyond oncology into autoimmune diseases and other diseases. And within autoimmune diseases, myasthenia is the first one that we're going after. Sure. Now, so multiple myeloma is another disease we're very interested in. We have two programs that are going after this blood-bone cancer. Myeloma is a very difficult cancer to treat and cure. There's been a lot of work in new therapies over the last decade. There's a lot of options that patients now have, but virtually every patient will eventually relapse. Our aim through the use of RNA cell therapy is to treat the disease early enough in a way that combines with the other therapies that are available to patients, such that we eliminate every remnant, every last cell in the body. And you can do that a lot easier when the patient has just recently been diagnosed compared to when the patient has the most advanced form of the cancer. It's true with any disease, and in particular, that's true with multiple myeloma. So the idea behind our programs is, let's make the therapy safe enough to go after patients when they are diagnosed early on, as opposed to waiting until the very end, right? So the two programs that we have for this indication are Descartes-8 for patients with newly diagnosed high-risk multiple myeloma. Okay, and these are patients that are recently diagnosed. They undergo their standard of care therapy, and we treat these patients at the end of their standard induction therapy and before their autologous stem cell transplant, to see if we can eliminate every last clone. And that clinical trial is underway. A recent addition is a program called Descartes 25. And the idea behind this program is we can use the cell as the factory for delivering a combination of highly potent anti-tumor proteins right to the site of disease where the myeloma cells are. So these cells are engineered, and they're mesenchymal stem cells instead of T cells, and they're off the shelf instead of autologous. There's some differences here, but they're engineered in order to go after the disease right where the disease is at, and then just deploy their cargo, a very potent, synergistically active combination of anti-myeloma proteins right where the myeloma is. So we're very excited about that. And that's in patients with advanced myeloma at first, and the hope and expectation is that we will eventually move that product into treating patients with early stage myeloma. The fourth clinical program we have is for respiratory diseases, and in particular, patients with acute respiratory distress syndrome. So ARDS is a relatively common and end-stage respiratory collapse that can be due to both infectious agents as well as non-infectious agents. Within the world of infection, before 2 years ago, the majority of cases were due to influenza, and you would have up to 180,000 such cases in the US alone. Now, after COVID, the vast majority of patients who suffer from ARDS suffer from it because of COVID. So it's a common condition. It's often what ends up killing you if you have COVID. And ultimately, there hasn't been a single new treatment that has been approved for ARDS over the last 2, even 3 decades. So there's an urgent need to develop new therapies. The way we come after this indication is to engineer off-the-shelf allogeneic mesenchymal stem cells that are engineered with a combination of DNases that work together to degrade something called NETs, neutrophil extracellular traps. NETs are these sticky webs of DNA that are expulsed by neutrophils in their attempt to control acute inflammation. And so they basically release these gobs of, just think of them as spider webs that are designed to entrap bacteria and viruses. But the problem is they overdo it. And as a result, these sticky webs of NETs serve as the nexus of inflammation. And so it stands to reason if you get rid of these, then you could relieve some of the blockage in the alveoli and allow the patient to breathe easier, and then eliminate some of this thrombosis in the venous system that blocks the blood from flowing freely. And so that's what this clinical study is about, using Descartes 30 to treat patients with moderate to severe ARDS. And hopefully the kind of safety data and preliminary efficacy data we intend to get will open the doors to be able to use the same product to treat patients with earlier stage disease, apropos of our ability to use RNA cell therapy to go after earlier stage indications and beyond oncology. So patients with pneumonia instead of ARDS, and even patients with other inflammatory diseases. In 2022, we hope to get some top-line data across 3 of our 4 clinical programs, which will guide our decision-making in terms of what the next steps might be. We have preclinical programs that are the next generation of the clinical programs we're currently in. And we're right now very focused on execution for clinical trials that are currently ongoing in order to be able to take a peek at some of the clinical data across our Descartes 8 myasthenia study, across the Descartes 8 in the frontline myeloma study, in Descartes 30 in ARDS, potentially even towards the end of the year, take a peek at some of the data for Descartes 25 and myeloma as well. So that clinical peak will inform the next set of decisions from a strategic perspective, and also inform what kind of programs we want to prioritize from a preclinical development perspective. I'm excited to be a part of this adventure in cell and gene therapy in general, and RNA cell therapy in particular. That the platform technology that we've built, that we've coined the RNA armory, is capable of generating more cells than has ever been historically possible, and engineering them with RNA that expresses itself over an extended period of time. And so it's an exciting time to be a part of this field and pioneering this new class of therapies, because in 2022, the technology and resources are available to be able to really make some cutting edge breakthrough technologies accessible to a large number of patients within the world of cell and gene therapy.